Hello everyone, my name is Shraddha and welcome to my YouTube channel Node of Biology. Today we will be discussing another component of the endomembrane system, the lysosomes. The term lysosome was coined by Christian Tadeu. This membrane-bound organelle is found in both plant and animal cells. It is absent in bacteria, where in place of lysosomes we have periplasmatic space playing a role similar to that of the lysosome. This organelle has a very high content of acid phosphatase and other hydrolytic enzymes. Lysosomes are the principal site for intracellular and extracellular digestion. They process undesirable material coming both from the exterior of the cell and waste that is produced inside the cell by degrading them. Because these enzymes work at a very acidic pH, sometimes they end up damaging the lysosome itself, which results in the destruction of other components of the cell, resulting in autolysis. And thus, the lysosomes are also known as the societal bags of the cell. Let's learn a few properties of the lysosome. Lysosomes contain 40 different types of hydrolytic enzymes synthesized by the rough endoplasmic reticulum. And all of these enzymes work at an acidic pH of around 4 to 5 and are known as acid hydrolases. Another important property of the lysosome is their stability in the cell. The enzymes of the lysosomes are enclosed within a membrane and hence are not readily available to the substrate. However, under pathological condition, the membrane can become labile and result in catastrophic consequences. Some of the key functions of the lysosome include intracellular digestion by the process of autophagy, extracellular digestion by the process of endocytosis, release of cellular and extracellular material during the developmental process. Leukocyte granules also have lysosomal natures, such as macrophages. It is also responsible in the fertilization of sperm and egg because the acrosome of the sperm, which is kind of specialized lysosome produced by the Golgi apparatus, contains enzymes such as protease, hyaluronidase, and abundant amount of acid phosphatase, and thus these enzymes create a channel through which the sperm is able to penetrate the egg. There are two types of lysosome, primary and secondary lysosome. In the primary lysosome, the first reaction, that is the acid phosphatase reaction, takes place. The secondary lysosome are of three types. They can be the digestive vacuole, which will result from phagocytosis or pinocytosis of the foreign material performed by the cell, or residual body, which is when the digestion of any particular material is incomplete and this particular substance then stays as a residual body in the cell, which is then eliminated via defecation. And lastly, autophagic vacuole, where the lysosome very frequently engulfs some parts of the cytosol by a process known as microautophagy. Now, some key terms associated with the lysosome include phagocytosis, endocytosis, autophagy, macropinocytosis, and exocytosis. Phagocytosis is a process by which unwanted material such as cells, debris, or any bulky particulate material is taken into the cell. Endocytosis is the uptake of material into the cell by invaginating the plasma membrane and then internalizing the engulfed product into a membrane-bound vesicle. Autophagy is the digestion of cytoplasm and worn-out organelles by cells' own lysosome. Macropinocytosis, also known as cell drinking, is a type of endocytosis that involves non-specific uptake of extracellular material such as soluble molecules, nutrients, and antigens. Exocytosis is the excretion of materials from the cell by vesicle fusion with the cell membrane, which can occur constitutively or can be regulated, such as secretions of hormone, antibodies, etc. For future videos, please like, share, and subscribe to my channel, Node of Biology.